Thank you so much, Camilla, for this nice introduction. So I can skip my part. There was just a small part of that introduction. I'm very impressed about how many people joined. Not sure if this should make me happy or nervous or both. So I welcome you all to my talk today about target trial emulations to study drug safety during pregnancy. And we're gonna, now it works. In the first part, we're gonna talk about some background, why it is actually important to do perinatal drug safety studies, and why observational data is a useful source to investigate further. And in the second part, we're gonna talk about the target trial emulation framework as a valuable approach to develop in this field. And it's gonna be mostly about considerations in developing a protocol for a target trial and its emulation in observational data sources. So I want to mention first that some content here is adapted from those excellent publications from Christa Huibrecht, Sonia Hernandez-Diaz, and co-authors in the US. They did excellent work, and I can highly recommend to have a closer look if you want to read more, learn more, know more. But first to some background. Some history. Some of you may have heard about the thalidomide or Contergan scandal. There was like a drug, a sleeping pill that also reduced morning sickness and was advertised for pregnant women in the late 50s and early 60s. And preclinical studies, they haven't shown pathogenic reactions in non-pregnant mice and rats. So there were actually severe consequences with like 1956, the first child born with a malformation. And by 1961, more than 10,000 of children with phocomelia, and only around 5,000 actually survived childhood. So it became clear quite soon that Contergan is a strong teratogen. And actually for the ones who want to know more, this is actually a nice documentary. You can have a look later. So the conclusions from this scandal was that the placenta was assumed to protect the unborn child from drugs, and that systematic monitoring of teratogenicity of drugs is required after drug approval. But later we also learned it's not only about teratogenicity, it's obviously a very, very severe outcome, but in perinatal pharmacoepi, we are also interested in pregnancy losses, in obstetric outcomes like gestational diabetes, or neonatal outcomes like small for gestational age, large for gestational age, but also childhood outcomes later in life. And one outcome probably most prominently discussed is neurodevelopmental disorders like ADHD, autism, but also infections, and the list could go on forever here. But where do we stand around 70 years after the scandals, and are medications safer today? How is evidence generated, and how many women actually take medications during pregnancy? So let's start with the last question here. And actually a big proportion of pregnant women take medications during pregnancy, either because they have underlying chronic conditions or incident diseases, but also obstetric complications, even complications in the fetus or conditions in the fetus. And our Swiss study in Swiss claims data, for example, has shown that across all age groups, more than 30% here, the pink part, of women take more than 5 medications during pregnancy. And this is not including vaccines or minerals, vitamins, or like those medications or supplements many or almost all women take. But then medication use during pregnancy is also increasing, and that's a nice study by a colleague in Denmark who showed that it's increasing throughout all age groups. And here also a statistics from Denmark about maternal age at delivery that is increasing, which obviously goes in hand with more chronic diseases that require treatment. And very importantly, actually a lot of pregnancies are unplanned. And that means drug safety during pregnancy is also a relevant field for women of childbearing age in general. And as many of you know, like pregnant women are typically excluded from randomized controlled trials due to ethical concerns. So official drug information generally advises against the use in pregnant women. However, there are occasional RCTs if the intervention is specific to pregnancy, such as preeclampsia. And there are actually ongoing discussions at the moment to when and why we should actually include those women. Like, is it really ethical to exclude them? Or is it also ethical to not include them? And I think they're going to be a nice following discussions also in the next years. There's also a new EMA guideline in 2025. If you're interested, you can look at it later in detail. I will show on the next slide. But then even if women would be included in those trials, they would in most cases be underpowered. So it's not even that useful for many outcomes. So there's a huge evidence gap at market approval of drugs. That's the information. You can have a closer look if you want. But then evidence can also be generated in animal studies. They're, however, poor predictors in terms of pregnancy. There is also case reports, pharmacovigilance that can give hints and clues, but also false alarms. There are some services across countries, one of them in Switzerland called Teratogen Information Service. But then why we are here today is the observational studies that are actually the largest source for evidence on drug safety during pregnancy, and very important. And I think I should quickly get like a small overview just to that we know we are talking about the same. So generally we can distinguish them in two categories, like prospective registries that are very common in pregnancy, like pregnancy exposure registries, very known in the field of anti-seizure medications. They collect data on a specific drug or a specific group of drug, here anti-seizure medications. But then there is also disease-specific registries that include information on a specific disease and different medications. And then healthcare databases, important for us here in Sweden, like the national registries from the Nordic countries, but also huge, large insurance claims in the US, electronic health records, for example, from the UK, but we could go on with examples here. And then very specific to pregnancy are actually, or not only in pregnancy, but very valuable in pregnancy are those multi-center and multinational collaborations, combining these sources to increase power. But that can obviously also be quite costly and complex. You need a common data model to make use of all the data. And there are some big initiatives in this field like Sentinel or the IMPRESS Consortium and Conception that do a big effort in this field. And then pharmaceutical industry is also actually providing data, like the FDA provided the authority to request pregnancy exposure registries as post-marketing requirement by the FDA. And there are similar trends happening in other regulatory agencies. So traditionally, as I said, the focus was mostly on teratogenicity due to historical reasons. And traditionally, we compare exposed versus unexposed, which at first sight violates several fundamental principles of causal inference. It's not necessarily bad. It can, especially if a teratogen is very strong, its evidence generated is still highly relevant, but especially for teratogens that might have a modest effect, it's obviously very important to have a robust study design. And that doesn't obviously mean that all traditional studies are not done well. There is a lot of good evidence generated on traditional perinatal pharmacoepi studies. But then in the recent years, there is a growing interest in conceptualizing causal inference based on observational data as an attempt to emulate a hypothetical pragmatic randomized trial and we call it the target trial. And I guess many of you, or the ones who worked in this field, they are very, very familiar with this. But just to have everyone on the same page. So the target trial is a hypothetical randomized trial that we would conduct to answer a causal question if we had no constraints, no ethical constraints, no time issues, no funding restraints, etc. And it allows the researchers to study how medications affect pregnant women and their babies, and here very important, at different stages of pregnancy without performing an RCT. We will get back to that. And here a short overview of such a protocol with the 7 components we need to define in advance, first in the target trial and then in its emulation. And yeah, this framework forces us as investigators to make each aspect of this protocol explicit, which is a big advantage of this framework. So it's a useful tool to guide us through the study design and to avoid self-inflicted biases like immortal time bias, selection bias, and so on. And very important, it allows us to identify unavoidable deviations from the optimal protocol, and making these deviations explicit makes it easier for us to interpret the results. And I think this is, especially in perinatal pharmacoepidemiology, very useful, because we have to sometimes deviate from the protocol, just how we include those women in the trials, and there is no way around. So this is a very useful tool in this regard. And I wanted to say a few words about how long these target trial emulation studies actually exist in the field of pregnancy, because many of us will know that the target trial has been around since quite some time, and has been, you can see here when you look on PubMed and you search for target trial, there are many studies since the 80s. And there was also probably things happening before that didn't call it target trial. And then we can see around, I mean, this is only limited to title and abstract, the keywords, led to around 600 publications. When we look at target trial emulation, it started to become prominent later with 500 publications in 25. But then you see here in pregnancy, the number is much, much smaller. And actually, most studies are from last year. And many of those studies are not even clinical studies. It's like methodological studies, and many of them from the US. But also, I had a look this morning, there were at least 2 Swedish studies, 2 Norwegian, a Danish, Canadian, Australia, also 2 African countries. But this is going to change a lot probably in the next years. Going to happen a lot of interesting things. And then here, just an overview about this framework that provides a structure to make each aspect of the protocol explicit. So from the causal question in step 1 to applying causal inference analytics. And today we're going to focus mostly on the causal question, and then on step 2 and 3, the target trial and its emulation, with a strong focus on eligibility and treatment strategy. But we're also going to touch upon the other components of the target trial. So an intuitive question, if we talk about the causal question, would be, if I use a treatment X during pregnancy, do I have an increased risk of outcome Y? And I mean, if the intention is not to show causality, this descriptive quantification could be good, but it does not provide insight what would happen without treatment. So we could ask another question: if I use the treatment during pregnancy, do I have an increased risk relative to if I have not used it? And obviously, we do not observe counterfactual outcomes. So this translates more into a question like, what is the average risk in a group of people like our patient who use the medication relative to the risk in an identical group of people who do not? But this is still not very adequately defined. It does not translate really in a testable intervention in a well-defined population. So the effects still depend on whether the patient initiated the drug before or after conception, what are the alternative strategies, etc. So this would be a better question for research, like, let's say, among users with certain characteristics, what is the risk expected for these treatment strategies? Like discontinuation of the drug versus continuation, or switching versus, or switching to a different medication versus no switching. And in this framework, there are actually many challenges for each stage of pregnancy that is very unique in this setting. And there are different treatment initiation scenarios. There is one preconception treatment initiation. Let's say you started treatment before pregnancy, and then you're interested in an outcome in the mother in the first trimester. But then you can also look at conception initiation, which is obviously very difficult because you often don't have really the at conception, and you wouldn't really aim for a treatment right at exception. So it's a bit harder. And then most studies, I would say, focus also on the post-conception. Let's say you're exposed during pregnancy, but it can also be exposed during trimester 1 specifically, or trimester 2 to 3, or by gestational wake, whatever you are interested in. Like here you can see the development of the fetus and embryo. And for example, you could say you're interested like in the first trimester when all the organogenesis takes place. You might be interested in all of them, so you would restrict to initiation in trimester 1, but you might also be interested in specifically eye malformations. So you would be very interested in week 4 to 5. So you can see here the sample sizes can get really small once you want to be specific. But even if you don't want to be specific, sample sizes can already be very small. And then in trimester 2 and 3, or towards the end of pregnancy, brain development happens. So most studies, studies that are interested in neurodevelopmental disorders in the offspring, like autism, etc., would be interested in exposures during the end of pregnancy. So now I would like to introduce you to an excellent paper from excellent researchers in the US, Christa Huybrecht, Brian Bateman, and Sonia Hernandez-Diaz. They put together a really nice paper in 2025 about this topic, and I can really highly recommend to dig deeper there. It puts together a lot of information that probably many researchers took into account, the ones who did well-performed pharmacoepi studies in pregnancy. But it gives a really nice overview and considerations you might not have thought about in the first place. And the focus is like a lot on US data. There is unfortunately not much yet done in the Nordic countries, although there is a big potential for this field also with Nordic nationwide registries. But obviously you can reuse a lot of information and adapt to different data sources. And they show a really nice figure 1 with the challenges for pragmatic target trial emulation in pregnancy. And you can go there and have a look what specific scenarios you're interested. As mentioned, we're going to focus today on eligibility criteria and treatment strategies, but we're also going to touch upon the other components of the protocol. And some recap. So enrollment into the study, the time 0, is when eligibility criteria are met, the interventions are initiated, and follow-up starts, and outcomes start to be observed. And this choice of this time 0 and the eligibility criteria should not introduce bias. So a core principle is that we selection into the cohort must be independent of post-baseline events, so after time 0, since these cannot be measured at the time of randomization in the hypothetical target trial. But this is often not possible in perinatal drug safety studies. So we often have to deviate here from the protocol. And in preconception studies, when we are interested in exposure before pregnancy, we often need conception as an inclusion criteria. So we condition on conception. And then for post-conception studies like exposures during pregnancy, delivery of the newborn, of the infant is usually, or most of the time, an inclusion criteria. So we also need to look in the future and condition on future events, such as, for example, live birth, but it can also be, let's say live birth for now. We go in detail later. And let's have a look in the next slides at post-conception scenarios. So one scenario is that we condition on the end of pregnancy at enrollment. That's probably the most common case. We want a delivery at the end. And as said before, database studies, including those in Swedish registers, they often violate this basic principle of not looking into the future to define this eligibility criteria by requiring enrollment for a minimum follow-up period. So most studies, as I said, condition on the end of pregnancy, because this is how we identify the cohort. Let's say based on codes for live birth, ICD codes, for example. But this results in several challenges. For example, if outcomes during pregnancy affect the likelihood of remaining in the healthcare system or insurance until the end of pregnancy, the outcomes can affect the probability of selection into the study. And this is usually no big concern in Nordic national registries because there is not really a dropout. You can emigrate from the country and you will be not followed anymore, but usually there's a very long longitudinal follow-up of the patients. But it's obviously a bigger concern in insurance claims. Many studies were performed in large US insurance claims, and people can change health insurance. Although in the US, people, or let's say in pregnancy, it's less of a risk. People usually stay with their health insurance during the pregnancy, postpartum period, or before pregnancy. And in the US, you also have this health insurances for pregnant women and elderly. I hope I'm not saying something wrong here, but there is also not a big risk for dropping out. But obviously in many cases it is. And a potential solution here would be to identify pregnancies based on the first positive pregnancy test or the first prenatal visit, and then censor at the date of the outcome, end of study period, end of enrollment, or end of follow-up. But how does this look like in practice? So in US studies, the start of pregnancy is often not known, and it needs to be estimated based on algorithms. There are also validated algorithms, so there was a lot of focus in the past on validating those algorithms, and other countries adapted these algorithms. For example, we used these algorithms in Swiss claims data and validated again, or ongoing validation studies are happening. And obviously, if you start, there is still can be low sensitivity and specificity of the estimated start of pregnancy. So you can miss pregnancies, but also have false positives. And as said before, that's less of a risk in these national registers in the Nordics, because you have that information. Then we have another scenario, conditioning on survival with the outcome until diagnosis. So let's say you have to survive and you need to be in the cohort as long as you want to observe the outcome. Let's say you want to observe congenital malformation, or you want to measure small for gestational age 1 day after delivery. And that means we introduce selection bias if pregnancy losses occurring after the outcome onset are associated with both the treatment and the outcome. And this can happen in different scenarios, 2 of them. One of them, for example, the first is when the malformation occurs among exposed pregnancies tend to be more severe and leads to a higher risk of, let's say, stillbirth or non-viable pregnancy in general. But also if exposed women are more likely to terminate the pregnancy because they got informed about congenital malformation in their fetus. And but we still need to restrict to live birth in most cases, or especially in the case of observing congenital malformations in the target trial and its emulation, because usually we don't have fetal autopsies that would give you information on malformations before the fetus was born. And yeah, that means prenatally diagnosed malformations are incompletely recorded. Then I think also very interesting, if we need to condition on the delivery, but also following outcome, that's actually also the case when we look at children during follow-up. And an example here is like offspring neurodevelopmental disorders. I mentioned before that the end of pregnancy is like the critical period for these outcomes. And so for the outcome to manifest, the child needs to survive for some specific time to actually get the diagnosis. So it needs to survive for several years in this specific case, because those diagnoses are usually not done at a very young age. Often happen after age 3 or 5, a bit depending on the country and the system. But fortunately, this is not of such a big issue here, because stillbirth and infant death are usually relatively uncommon. So there shouldn't be too much worry about that, but still something interesting to think about. And then it's also, we can also talk about conditioning on access to infant records. So in US claims data, or in general in claims data, they sometimes need to restrict to those mothers and babies that can be linked, because the outcome is usually, if we are interested in outcomes in the child, it's recorded in the child records. So they need to be linked, and this can obviously lead to selection bias if not all mothers and babies can be linked. And I mean, a solution to this is to evaluate whether the association, whether the restriction is differential by treatment, and compare the characteristics between pregnancies with and without linkage to the newborn. so we can adjust for common causes of the successful linkage and the outcome, and assume conditional exchangeability within levels of covariates. And again, here, this is not of a big problem in the Nordic national registers, because we usually have complete linkage between mothers and babies. So we can go on to restriction based on the indication. I think that's a very interesting point. Because most target trials or most RCTs also would only randomize patients with a specific indication to avoid indication bias. And that's arguable when studying drug safety. There is, for example, a study on psychotropics and birth defects. I will get into that more on the next slide. But also arguable when studying unintended benefits, like metformin for cancer prevention. And obviously, like restriction to a specific indication is a very useful, transparent, and effective way to prevent confounding by indication at the design stage. I think that's what most of us are very familiar with. But we can also do that at the analytic stage, like through matching, standardization, or model adjustment. And then when could this be useful despite a clear deviation from a realistic trial? So it's if the indication is not a risk factor or a proxy for risk factors for the outcome, or if the indication is not well captured. For example, when we look at teratogenicity and psychotropics, both restriction and adjustment have led to successful confounding control. I think that's a very nice study. But then we go over to treatment strategies. So just a small recap. So these are initiated at baseline at time 0, and it can mean initiating medication, but also discontinuing, switching, adding a medication, reducing the dose, increasing the dose, etc. And yeah, in many studies, the exposure definition does not define treatment at baseline. So patients exposed at some point during trimester 1 are often compared with unexposed patients with focus on the etiologically relevant window for malformations. And these traditional comparisons, they do not answer a causal question, and they look into the future to assign subject to treatment groups, and that can introduce bias. And let's discuss now how target trial emulation can help. And one scenario here, again, in the post-conception initiation phase, we can compare the risk of spontaneous abortion in those vaccinated and those not vaccinated during the first half of pregnancy. And here you can see spontaneous abortions by gestational week. They mostly happen in the first half of pregnancy. And overall, they happen in like 12 to 15% of pregnancies. And an intuitive comparison would be, for many researchers, would be we compare patients who are not vaccinated, the blue ones, at start of pregnancy, and then we look at those who are pregnant and get a vaccine, let's say in week 10, and we compare them. But as many of you know, this can introduce, or will for sure introduce, immortal time bias. So there is a shorter opportunity for the outcome from vaccination than from conception. So quick reminder, time 0, when this is when 3 things happen. When the eligibility criteria are met, let's say the woman is pregnant. Second, when the treatment strategies are assigned, they're vaccinated, and the study outcomes begin to be counted. here spontaneous abortion. And then one option would be to set time 0 at the time of vaccination, because the woman is pregnant, you get the vaccine, and you start to count outcomes. But that obviously means that there are many different time points of vaccination possible during the first half of pregnancy. So you need to do like sequential trials, many trials, each week of follow-up, and the time 0 will differ in each trial. And you would include in each trial the eligible individuals at its corresponding time 0. And then you would combine all those target trials for a more precise estimation. But then you also have another option. You could also set the time 0 at start of pregnancy. So the eligibility criteria are met, you're pregnant. Second, you're not yet vaccinated, so this is a problem. But then third point are also fine, you start to count. So how do you do this if the vaccination is not known yet, the time point of vaccination? So like in other pharmacoepi studies, that's not unique to pregnancy. You can use a cloning, censoring, weighting approach and clone each subject, contribute to both strategies, censor them once they deviate from the strategy, and then apply weights to adjust for the informative censoring. Let's maybe skip this one. I think this one is also very interesting, the active reference group, because the target trial emulation framework, or in general pharmacoepi studies, they highly support the use of an active reference group, but is that really feasible in all pregnancy studies? And I think that's a very interesting point to think about, because in pregnancy specifically, no treatment could actually be an acceptable treatment strategy. And it is possible to restrict or adjust for the indication. But the problem here is if the indication has different disease severities, there is a concern in the non-users, and that can obviously introduce confounding. But it's very challenging in pregnancy studies to identify a good active comparator that is widely used, where safety data are available. Actually, that's very important, because imagine you have a medication as comparator that is causing malformation itself, but you don't know it because there is no data. And this can actually happen quite often, that we don't know if there is a modest increase. So you would kind of dilute the associations. Therefore, a lot of pregnancy safety studies actually study the safety of a drug versus no pharmacological treatment. And for sure, if confounding by indication is no concern, then the unexposed comparator group may be reasonable. But since there is no placebo, neither in the pragmatic trials nor in emulated trials, we are making assumptions. So we assume that they have comparable healthcare visits, comparable access to care that may affect the diagnosis and the outcome. And we need to make these assumptions plausible by balancing such factors. And this is actually very relevant in women's health, because there is also, maybe not very relevant in this field, but if you compare men and women in one pot, there is often an issue that women have different healthcare utilization than men. Let's check the time. Okay. I have some additional comments. And I think since we are here in Sweden at KI, I wanted to mention some potential for the Nordic nationwide registers, because they have not yet been used as much in this field, although it's emerging and a lot happening. But I personally think there is great potential because of quite good information on the start of pregnancy, which is very important, just to highlight again, because gestational age is such an important component when studying pregnant women. Then we also have very good mother-baby linkages. And there is also a lot of multinational collaborations within the Nordics that are used to work together, have similar data structure systems. So it's a lot of great studies emerged from such multinational Nordic collaborations. In the best case, obviously, with also countries, with other countries. And then there is also a long tradition of collecting this longitudinal data over decades, and that the registers are nationwide. And now I can speak maybe only about Sweden because I know this most in the Nordics. Also the quality registers that even enrich with further, sometimes very specific information on different diseases or conditions. And these registers, they are all linked via the unique personal number. Maybe other Nordic countries have other names for this number, but that's really unique. And then before we go to the conclusions, we now talked about pregnant women, but obviously also children and older adults are very valuable when using the target child emulation framework to study drug safety, because they're often, like pregnant women, underrepresented in RCTs. Children are included if the indication is relevant for childhood, but otherwise often excluded, also ethical concerns. And older adults are also often excluded, or in general, polymorbid patients with chronic kidney disease. For example, we did a study in Swedish register data on acute coronary syndrome in older adults in terms of major adverse cardiovascular events and bleeding, because they were not represented well in the RCTs. And this is even a field with big RCTs and many affected patients, but even there, they were underrepresented. So let's go to the take-home messages. So great attention has been given to confounding and misclassification when discussing the validity of observational studies over the past decades. So the new focus on target child emulation also emphasizes other biases resulting from temporal misalignment between the fulfillment of study eligibility criteria, treatment strategy assignment, and the start of follow-up. And these target child emulation studies are a valuable alternative or complement to RCTs, as pregnant women are typically excluded. And when included, sample sizes would usually be too small. And then the TTEs forces us to ask the causal question, to clearly define the population, exposure times zero, the outcomes, all the aspects, and make deviations explicit. However, this framework is not a magic solution. If we can control self-inflicted biases, but obviously not data problems we cannot solve. But yeah, sometimes we have to deviate, and just due to the natural history of pregnancy and characteristics of observational data sources. And I would say TTE in pregnancy is a rapidly growing field with emerging evidence, new methodologies, fruitful collaborations across different countries. So I think the next year is going to be really interesting. And I think there are excellent researchers in this field. And yeah, I'm really curious how this is going to develop over time. And I made a slide for some further reading if people want to have a look. And then I would like to thank you for your attention.